All right. Hello, good morning, everyone, and welcome to my talk about the comparison of cecal drop microbiota between two selected lines of laying hens, selected either for high or low occurrence of feather pecking. So for those of you who are not too familiar with the issue of feather pecking, it is a very serious welfare and production issue in modern commercial egg production. Because it can develop into cannibalism and then result into very high rates of mortality of up to 40%. It is a bird-to-bird behavior where one bird pecks at the feathers of another bird and removes the whole feathers or fragments of feathers. And it also has a genetic component to it, and that's how those genetic lines were created, either selected for or against this behavior. And those are also the birds that we used for our experiment. And interestingly, feathers are also often ingested in the process of this feather pecking, and that you can see here in this gizzard. I hope you can see, it's very dark, but this gizzard contained a very significant number of very large feathers in just one single chicken. So why would chickens eat feathers, or why would any bird eat feathers? It sounds very unnatural. However, we know that feathers have the capacity to increase the speed of feed passage. So the ingestion of feathers makes food travel quicker through the digestive tract. And there has also been a connection found between the occurrence of feather pecking and the occurrence of intestinal parasites, in this case worms, in laying hens. And that makes sense if we look at wild bird species. Wild bird species who purposely eat feathers in order to create pellets in their digestive tract and then eject those pellets to get rid of buildup of intestinal parasites. So one of the beneficial effects of feather eating might be to control intestinal parasites. We also looked at organisms that live in the gut of those high and low feather pecking chickens, not necessarily harmful ones or parasitic ones, but we wanted to know if this selection of birds for or against feather pecking would influence their microbiome overall, so whether they would have different microbiomes if they were selected for or against feather pecking. And for that we chose to go with a feces sample instead of the common euthanasia and then sampling the cecum of the birds, because this would only allow you to sample the bird one time and one time only. And by sampling feces, you can sample them for multiple times. And our hypothesis was that the microbiome would be impacted by the genetic line. So what we did very briefly was we took 20 birds selected for feather pecking, and we took 20 birds selected against feather pecking. We placed them in single cages. They were of the 12th generation of this selection experiment. And then we collected the cecal part of the feces, stored it, and then had it sequenced by our collaborators at McMaster University. For the results in alpha diversity, we didn't find any differences between the two lines of laying hens. However, beta diversity, we found significant differences in this measure of dissimilarity between two populations. And that was despite the fact that those birds were housed in identical environment and given identical nutrition. If we look more closely at what those differences were, we found a very significant increase in Clostridia present in the high feather pecking birds, and we found decreased presence of lactobacillus, for example. And if you're wondering why were there more Clostridia in feather pecking birds, many people would probably guess, oh, maybe they were sick or maybe they had diarrhea or something, because Clostridia are often associated with disease, but those birds were completely healthy and they were not the kind of Clostridia that cause a disease. However, it is very important to note that Clostridia are known to be involved in the metabolism of keratin, and it only makes sense that in an organism or environment where you have keratin readily available, that these kind of organisms would thrive better. And this was very interesting for us because we previously looked into the metabolism of amino acids, and we know that Clostridia, among others, is a significant part in the metabolism of amino acids. So we want to bridge our knowledge from what we know is going on in the physiology of the bird, and we want to add to that what is going on in the metabolism of the microbiome, and that could be very well impacted by metabolic pathways such as the tryptophan metabolism. So in the end, we would like to assess whether feather pecking can be induced by a condition that has to do with the tryptophan metabolism, and whether the microbiome plays an important role in that kind of development of the disease. And ultimately, we would like to know if feather pecking does in fact constitute a microbiome dictated health problem, and that would then allow us to counteract feather packing by identifying nutraceuticals, for example, to prevent feather packing or treat it in practice. And with that, I would like to conclude and thank all my collaborators and helpers in this project. Thank you very much for your attention.